Hello, and thank you all for joining today's Science, Conservation, and Humanities lecture with Dr. Dennis Stevenson, Senior Curator Emeritus and Cycad Specialist at the New York Botanical Garden. My name is Charles Zimmerman, and co-hosting with me today is Dr. Barbara Ambrose, Director of Laboratory Research and Associate Curator of Plant Genomics at NYBG. At any time during today's event, please feel welcome to type your questions by clicking the Q&A button at the bottom of your window, and our host can raise these for our speaker to answer at the end. Live captions can be enabled by clicking the CC button, then show subtitles or view full transcript. As a reminder, this presentation will be recorded and shared in the NYBG lecture library. Now, please welcome Dr. Barbara Ambrose, who will introduce our speaker for today.
Good morning. It's hard to imagine that Dennis Stevenson really requires an introduction. He is the biggest and most impactful advocate for botany that I know. In a few minutes, it will not be possible to detail all of his accomplishments. If you want to know that, I suggest you go to his CV on the NYBG web, but here is just a brief review. Dennis Stevenson got his bachelor's and master's from Ohio State University with Richard Popham, his PhD at UC Davis with Ernest Gifford, and had postdoctoral fellowships with Barry Tomlinson and a NATO postdoctoral fellowship to Kew. He has an honorary doctorate from the University of Naples and enjoys being a professor doctor. He has been at the New York Botanical Garden for nearly 40 years. Before I get to his research, I just want to point out that Dennis is innovative, not just in botany, but in fundraising for NYBG science. And he has always been an institutional thinker. The amazing modern laboratory facilities and programs we have at the New York Botanical Garden would not have been possible without him. Dennis has researched every major land plant lineage, and he has always incorporated the latest tools. Today, he will tell us about his research with Cycads, and I thought it would be easy to summarize this work, but it's not easy because he has done an extensive amount of research on Cycads. Dennis's first NSF grant in 1978 was on the comparative morphology and development of vegetative and reproductive shoots in psychedelics. He had many more grants to research Cycads and many other topics in the intervening years, but it is telling that his most recent grant submission is an NSF Opus submission on Cycads. Dennis has more than 350 publications in total, and of those, more than 165 on Cycads. His first publication on Cycads was in 1980 on the radial growth in psychedelics, and his most recent publication on Cycads is currently in press on observations on vegetative branching in Cycads. Dennis has authored floras involving Cycads and has several works and agreements to protect Cycads covered by CITES and the IUCN since the 1980s. I think many people in this webinar are here because Dennis has impacted their life and career in some way. Dennis is generous with his time and knowledge, particularly with students. He has the most encyclopedic knowledge about botany than anyone I have ever met. I, for one, am extremely fortunate and grateful to have Dennis as a colleague, mentor, and friend, and surrogate grandfather to my son. Thank you. Dennis, I'm looking forward to hearing about your comprehensive and continuing research on Cycads.
Well, I think it's actually time to stop after that introduction. I don't know if I can do any better than that. What I'd like to do today, I'm not going to talk about systematics very much in this lecture, in the sense that most of the audience are systematists, at least at the garden, and you can read about those things in floras and whatnot. What I want to do is just track the way a career might develop and how one might, through time, look at science endeavors as problem solving and how the solutions to those problems can often inform us about biology and also about systematics. I should point out that most of the work in here has been done with collaborators. Some of it is, my early work was just me, but in the modern days, we publish with many, many co-authors. Single-authored papers are very rare, and I realized that recently, and my most recent paper that Barbara mentioned is actually single-authored. So to get started here, it's just sort of a path if you look at this trail, and we're going to go through each of these topics in a little bit. So I just start out with the systematics part, and you can read my trees here, and we'll just move on from there. So my whole notion is, as I said, evolution is also about problem solving. And so we want to look at how those problems are solved and the various ways those things might come about. And one of the issues that often appears in systematics is this concept about homoplasy. Well, as I say here, in reality, homoplasy is just our ignorance of what was going on. And it really doesn't exist. It's just because we've misinterpreted things and we think of it in that way. And one of my examples in molecular systematics is here. An A is not always an A because it's not the same A if it was a T for a while when you track it on the tree. So we only know it's not the same A because we're able to develop that in a historical concept. The other thing that it does do for us, under seeing homoplasy, is it sets the stage for what people like to talk about as reciprocal illumination. And we'll see examples of that. So all of the work for us starts in the field. And sometimes that can get a little intense. Like if we look in the frame in the upper right, we start in our upper left. We start in the lower left and we have to walk all the way to the frame in the upper right. But when you get there, you're rewarded by finding these magnificent trees that have been living in China since the Permian. Other things are like to get people to lower you onto a cliff face so you can find a species that was only known from a type and had never been seen since. And so you take those discoveries and you bring that material back. And often you bring back living plants or your herbarium material. And in the case of people with an anatomy morphology background, the good old pickles. So we bring the pickled stuff back and we look for structural features that help us understand how these plants are put together, how they function, and how those big trees can stand up. So here's some lists of things that are peculiar only to cycads, are not known in any other group of plants. And then inside the cycads on the right-hand panel, there are apomorphies for various groups inside of characters that are also not known in other groups. So this becomes a basis for us, and we can take this information and we can expand it to more and more species. So here's some examples of the concentric steels in the upper left panel and various things, cone domes in the center panel on the top, the omega pattern to the leaf traces in the lower right, the girdling leaf traces, and etc. So we can obtain this anatomical information actually in a pretty simple way. These things I got because there was a hurricane in Florida that knocked over some big trees because the horticulturists don't want you chopping up these rare plants. You can base a lot of things just on simple field observations. For example, these notions of branching in the cycads. And the cycads do not have any axillary buds like the rest of the seed plants, but rather they branch dichotomously. And you can see that in, for example, the upper left, you have a dichotomy and then a subsequent dichotomy on the right-hand shank in a perpendicular plane to the original one. In other words, this is like a giant psilodon plant. This is not a very good way to grow because it structurally becomes very difficult to stand up. So they really don't do a lot of this. And you can demonstrate that those are dichotomies partially with what I call slice and dice botany. You get your machete out and a little bottle of stain, and you go whacking the trunks up, as we saw in one of the earlier slides. And in this case, you can see that one stele in A, actually, if you look at it serially through the stem, turns into two. And if you keep going to the top, in H, you'll see that it branches at a right angle to that original place. So we can demonstrate this notion of the dichotomous branching through simple anatomy. Now, another set of characters that are interesting in this group of plants, which I found rather interesting, is their chemistry. You know, we always used to call this blender botany. You just get your blender out and grind some stuff up and do chromatograms of whatever you're going to do. There are two types of compounds on the left there, the MAM glycosides, and then this BMAA, which is a non-protein amino acid, that are unique to these cycads. They both cause problems in organisms, and we'll see that in a moment. The other thing that was really interesting was to look at, they have mucilage, which is actually a polysaccharide. And you can hydrolyze those polysaccharide into sets of monosaccharides. And what's really interesting here, if you look at Cycas, for example, versus Encephalartos, each genus has a unique profile of these monosaccharides when you hydrolyze that. And it became great markers for identifying genera. And interestingly enough, for CITES enforcement, when people take off all their recognizable things, we could use this character to say, look, you've got an appendix 1, encephalitis is all appendix 1, you're not passing go, you're going to jail, and the plants are going back home. Now, the other thing you can do when you have a lot of the structural data is you can begin to look at fossils. And these speak for themselves. We have the fossil stuff usually in black and white here, and the living ones in some kind of color. And if you look at the top middle where it says extant, that's Encephalartos. And if you look behind my picture at that fossil, you'll see that flap on the left hand of the extant that's in the fossil. In fact, it's absolutely obvious. And the other one is that seed with that thing called a coronula in the middle of the seed, which sort of bursts open when it germinates. And you find exactly the same structure in that fossil from Argentina on the right-hand side of that. So we know, and then the Cycas revoluta sporophyll there is basically almost identical to an early Permian fossil in the upper left. So we have very good morphological characters, and we actually have good preservation to be able to build things on. Similarly, some of the anatomical features, like the omega leaf trace in the upper left, the middle panel is a fossil cycad from Argentina. It's Wintucycus. And on the right-hand side is actually a psiloidin block that belonged to Charles Joseph Chamberlain. In the lower panel, cycads have this weird thing where the protoxylem transfers from a exarch to an endarch thing as the leaf traces depart. And we can actually see that 17 is an extant, and C and D there are actually fossils that do exactly the same thing. So we have all kinds of neat characters to look at. Interestingly, this Zamia on the left, nice cone there that I had in the jar, and A is a fossil called Beania, which was supposed to have a pendulous, perhaps megasporangic cone with these wide internodes and a lax structure. Interestingly enough, when your pickle collections dry out, you can make all the Beanias you want and give them to all your paleobotanist friends. The other thing that happens with fossils, if you note here in this panel, in the bottom, these holes in the walls of the epidermal cells. And these perforations actually were first discovered in fossils, and then they were seen in the extants. So whereas the extants can inform you about the fossils, equally so, the fossils can inform you about the extants. If you take this morphological and anatomical data, you can do a phylogenetic analysis using parsimony or whatever you want to do, and you get some sort of tree. And because the fossils are so rich in some of the anatomical and morphological data, you can add the fossils to that. In this case, we have more fossil genera than we have extants, and we have fairly reasonable resolution to this. And it allows us to place the fossils with the extants to understand something about when features occurred in lineages through time. So it gives us a time frame. Now, there are some kinds of characters, as you recall, I mentioned homoplasia at the very beginning, which is supposed to be the big bugaboo, but I actually like it. A good example in this cladogram is megasporophyll shape, where the megasporophylls are sort of flat, if you know diunes or even cycas, and others that are peltate, like the Zamias or that Encephalartos that was shown earlier. And what you see here is 3 instances of the independent development of megasporophyll shapes. Now, there's an alternative interpretation where there's a gain and a loss, but we'll use this, this analysis this way. And let's just take a look at that now. Well, if we look at these cones and we look at Encephalartos ferox or Macrozamia, there's a stalk from the center of those megasporophylls that go back to the cone axis. If we look at that in Zamias, it's the same thing. There's a Zamia cone on the left. If you take them apart, you see the stalk with 2 seeds on either side, or if you look at the end, it's like an umbrella. In fact, one might describe that as, I describe it as peltate. So let's take a look at these. Well, it ends up the Zamia clade, the stalk is a post, and it develops a ring meristem that allows it to expand in this peltate shape, and then the ovules develop. Interestingly enough, in the Encephalartos clade, those peltate megaspores develop their peltation because of the compression of the cone. So they start out flat and they develop an adaxial meristem, and then the bottom protrudes down because of compression of the cone, and you end up with this peltate sporophyll. And the really cool thing about this is the vasculature of the Zamia type is three-dimensional. The vasculature of the Encephalartos type is actually in one plane, like a flat sporophyll. So from that, we know that in the panel on the left, this is not, it was my ignorance of not how to code these, but by looking at it and doing reciprocal illumination, I do now know there's at least two different origins of these things, which is behind my picture there, but it's megasporophylls, flat, adaxially thickened, or truly peltate. Now, Bauhinia, interestingly enough, I never had enough material to know what was going on in it, but I do have it now, just a matter of not being so lazy and getting in there and sectioning them. But it means when I section them, I can look at the vasculature and I can look at the development, and lo and behold, Bauhinia may actually move somewhere else in the tree, which would be more in keeping with some of the molecular results that we get. Moving on, other neat questions were this. So when I was a graduate student, everyone went around and did what we called Flyspeck Botany. That was counting chromosomes. So, that's what you do. So we did a lot of that with my Italian collaborators. And what I really want to, the point I really want to make is look at those diploid numbers, 23, 25, 26, 27, 28. This is all in a single species, and some of those different numbers would occur in different populations. And what you can see is if you look at the N number, they can actually all breed with each other. What is going on here with the changes in chromosome numbers? And I will point out that the arm ends are always the same number, no matter whether the number is 23 or 28, the number of chromosomes are. So that has to do with metacentrics versus telocentrics. So let's take a look at Robertsonian fissions and fusions. Here's what happens when you get fissions. Up in B, you divide the metacentric into two telocentrics, and you increase the chromosome number by 1. Conversely, if it's Robertsonian fusions, you take the two telocentrics in C, fuse them together into a metacentric that's in B, and you reduce the number by 1. So the question was always, what is going on? Are we having fissions and fusions, or perhaps both? Well, if we look at this in terms of a phylogenetic analysis we did some years ago, and this topology's changed slightly, but for our purposes here, it makes no difference. We will see that the 16 is a basic number in all the major clades here. And so we would infer from that that there's increasing numbers through time in these groups, and therefore fissions are what is going on. I will tell you that getting the material for that Zamia that I showed you, we had to take a boat in the middle of the night to Tres Marias Island in Mexico to get them at the base of the prison walls. That's field work. Let's go back to chemistry for a moment. So there are these compounds called cycosine or macrozamia, that whole group we talked about. And one of the interesting things about those things is these are highly toxic to most organisms. So if we were to eat them, that compound would become cleaved and then would become toxic and cause cancers, etc. So this really prevents herbivory at a certain level. However, these butterflies, Humaeus, they love these plants, and you can see their eggs on the cone there in the center. And then you can see their larvae and pupae in the lower panels and the adult butterfly. They munch on these things. It's an endangered species, that is the butterfly, but so is the cycads. So Fairchild had a huge problems with the butterflies eating all of the endangered cycads, but they were endangered butterflies, so you had to let them do it, I guess. But the point here is these butterflies apparently are able to take these compounds, and they have a bacterium or something in their gut that, when it's cleaved, put it back together into a non-toxic phase. Shayla has done a lot of work on this recently, so I'm not going to go into it. She can tell you about it. The other compound is this thing called BMAA, or the non-protein amino acid. And this thing is pretty cool too. Non-protein amino acids will actually be incorporated into a protein, and consequently, when you consume them, your protein will have the wrong configuration. And so for insects and things, that means they can't get through their life cycles. they would be sort of monstrosities of some sort. The other thing BMAA does, it's what's called a glutamate agonist. Now, this is a unique compound to cycads. I will tell you, there's a compound called BOAA in lathris, in a legume, that also causes neurological problems. So with the framework, let's kind of look at what's going on, because this involves pollination biology too. So early on, you always read that cycads were wind pollinated. Now that yellow arrow up there in the upper left, if we dig all that stuff away and we see the upper right panel, there's a cone down there that's been pollinated. And there aren't hurricanes every year to do this in Florida. This was actually in Miami at Fairchild Tropical Gardens wild area. And this cone was good and it had good seeds. And we were pretty sure the lizards weren't crawling around in the leaf litter doing the pollination. So Knut Norstog and I, and it's just one of these things that I would call serendipity botany or serendipitous botany. And that is, we started looking at these cones because I was collecting them to look at ovule development, something that I've never finished, but, and trying to work on with Cecilia. We have here, we noticed that the cones would open for a day or so where those red arrows are. At the same time, Knut was doing something with pollen and collecting the pollen cones. And when we looked at the pollen cones, you'd see these weevils, the green arrows in the lower left, all over the surface of the cone. And if you look at the cones carefully, where you can't see this there, but there are holes in the ends of those microsporophylls. So we took young cones, like center panel green arrows, we put them in beakers with something over them, and all these weevils emerged. So we're beginning to go, aha, perhaps there's some kind of insect pollination here. So we did exclusion experiments like you're supposed to do. It took us 4 years to do this because the weevils would actually chew through the nets, and you see rubber bands on those because they would come in underneath, etc. But with perseverance, we were able to work out this life cycle. These weevils oviposit in male cones as they're emerging. They then emerge as adults, and they do two kinds of things. They go to ovulate cones while they're open for a day and run around inside. And what they do in the ovulate cones is they actually copulate. They then go back to male cones and oviposit into those young male cones. And in doing so, they carry the pollen back and forth. They can see the eggs there. And they also, there's these things called gold cells. Now, gold cells you can see with a razor blade, but gold cells are actually the place where BMAA is. You can demonstrate that through histochemistry, etc. So gold cells are concentrated with BMAA. So you have to say, these guys are eating on these cones. Why doesn't this BMAA as a non-protein amino acid keep them from going through their pupal stages and whatnot? Well, let's take a look here. If you look at the gold cells there in fresh tissue, you can see they're gold. You can localize for BMAA. You see it all over the place. The really interesting thing is that in the male cones, the cells with BMAA never disperse the BMAA out into the tissue. In the ovulate cones, as shown in the lower two panels, they disperse it into the tissues and make the total tissue toxic. If you go back and you look at the weevils, and that's shown here again, the weevils actually pass those gold cells intact through their gut and their system and excrete them into their cocoon cases, making them toxic to predators. The other thing these things do is a lot of the male sporophylls fall on the ground and the weevils stay in diapause. So the next year, when the cones emerge, they're released from diapause. Again, an accident in the lab where we were trying to draw these, and the artist came in one morning and all these cones had weevils coming out of them. What I would say about that's important about this is that there are only cones around for 2 or 3 weeks of the year. So the weevils have to do something for the rest of the year. So this diapause mechanism takes care of that. There have been many studies on cycad pollination since that time, and they've all shown that almost all of them are insect pollinated with various kinds of insects and not just weevils. Well, BMAA, when you get to mammals, is called a glutamate agonist. And this is not a good thing for you to go around eating these. Glutamate agonists act as an agonist against glutamate in your glutamate receptors in your neurobiology system. And you can see the similarities of the compound. So the way this works is that's just a figure of a neuron with some glutamate out there as a neurotransmitter. You get the signals, and then when the neuron fires, the glutamate's released back into the pool. If you put BMAA in there, it goes to the receptors, it stays there forever, and the neuron just keeps firing and it dies because your neurons have only a limited number of firings in your lifetime. So the idea here is then, how does this thing work? And so let's take a look. Well, I think I know what happened to the dinos. They ate too many cycads. But through people at NYU, particularly Gloria Carusi here, we looked at this GluR blocking drugs system in Arabidopsis because she is interested in glutamate and glutamine and nitrogen fixation, and these things are involved in that. And one of the things that happens easily here is when you grow Arabidopsis seedlings in the presence of BMAA, you get the plants on the left with the long hypocotyl, little funny cotyledons that never really open up, etc. And if you do BMA and that's in a dark grown plant. If you put BMA in there, you get basically the same syndrome as we see on the right. So BMA actually works as a glutamate agonist in plants, we thought, perhaps. And we labeled the glutamate receptors that we know are in plants. And lo and behold, they're actually located in the sieve cells along the veins in the leaves and the rest of the plant. We're going to go somewhere with this. A gene tree, as it shows here, suggests that all organisms have glutamate receptors and that there's these three classes that it says. I would point out here that we probably share 70% of our genome with Arabidopsis. Think about it. The Krebs cycle, cell division, all of these sorts of things are shared across all living organisms. And apparently glutamate receptors are another one. So the idea here is that there are these conserved amino acid sensing things, as we say here. And glutamate receptors can affect us in our GluR receptors. So one of the things that were done was to look for BMAA resistant mutants through screens, which were done and found. And it ends up that there are Arabidopsis mutants that are resistant to the Cycad produced BMAA agonist. So we took those things, actually Gloria did this, and she did something with it. Before we go there, let me just summarize this. So we know everybody uses it, we know it's in everything, and we know it's in the plasmolena. Now you have to ask yourself, if it shuts Arabidopsis down so that it can't grow normally, how can Cycads, which produce it, not be affected? So BIM mutants, we think, has implications for treatments in neurological disorders, such as Lou Gehrig's disease. And this was all partially pushed to me by my friend Oliver Sacks. So one of the things that was done was to take a xenopus egg in a solution, and you can measure membrane electropotentials. And if you put BMAA in there, you actually shut down the electropotentials to naught. But if you insert the BIM mutant gene into that membrane, you get about a 70% recovery. So there may be something that people can do with it. And why am I going into this? Well, there's this thing called Guam Dementia, and it deals with Cycads and the fact that people in Guam were coming down with neurological disorders precociously. They'd be like 90-year-old neurologically when they were 40. And people knew that they ate Cycads, so they always blamed it on eating the Cycads. But as you note there, you'd have to eat about 8 kilos of Cycad a day to get this disease. So what was going on? Well, fortuitously, Oliver and Paul Cox discovered, well, two things were discovered at the same time, more or less. The fruit bats in Guam actually eat the Cycad seeds, as we see there. And the incidences of Guam dementia on Guam in both male and female people went down to 0 by around the year 2004. And simultaneously, the flying foxes went extinct. They went extinct because the people ate them, as you see in the upper right, and they ate them whole. So the idea was that there was bioconcentration. Big problem. There's no fruit bats to go check whether they really have a bunch of it. But, and I sold this to someone at NSF once, the sub-head of NSF, who was a neurobiologist. They were able to demonstrate the bioconcentration because of the museum specimens around the world of the flying foxes, and they could check the skins of those things, and they found a 4,000 meg bioconcentration. So now we have a good cause and effect, and this lets you know what a collection can do that you never would have suspected. And what the people in Guam do, because they love the bats, they now import fruit bats from Australia. Tasty. Where does this BMAA come from? Well, there are two sources. These so-called agiotrophic coralloid roots in the cycads, and these are cyanobacteria that are nitrogen fixing. They're members of the Nostoc group and Annabeta and whatnot. And the interesting thing about that is, and that's this zone in there, that's that blue-green color from, quote, the blue-green or cyanobacteria. Anyway, you find in the root cells a lot of gold cells with BMAA. And interestingly enough, generally speaking, plants use glutamine in their nitrogen pathways, and so do the bacteria, but when they're together, they actually use a different pathway, a citrulline pathway, which I don't know any more about, but Gloria would. So let's just summarize a little bit here of some ideas. So we have some new discoveries about BMAA, which has long been known in the cyanobacteria as a neurotoxin. And how did the cycads get them? Well, one thing we know is the gold cells in cycads are actually found mainly near the shoot apex. And as you go down through the stem into the older parts, you really don't find them very much. It becomes dispersed. So the idea was a hypothesis for the Guam Dementia was the first level of bioconcentration in the cycads from the cyanobacteria and then into the fruit bats. The data shows, though, the concentration is too low for the only source. And plants that never had the symbiont, in other words, never had coralloid roots, and that you can monitor that when you grow them, also contain BMAA and have gold cells. So where did that BMAA come from? And the idea here is that possibly there was an ancient horizontal gene transfer, similar to like the one of neochrome. And I'll say this, every cycad that has ever been looked at, the living ones, has BMAA in it. Now, moving on to different things and doing different things with the plants, these are all endangered species. They're all known to be, are CITES listed, mini CITES 1 or CITES 2, and people smuggle them because some plants go for like $50,000 as a small seedling. The good one is that plant in the lower left, Microcycas. That was a male plant, the only known female outside of Cuba. At Fairchild, made a ovulate cone one year. I pollinated it, got about 3,000 seeds. And Bruce McAlpin, head of horticulture here at the garden, was offered $3,000 for one of the seeds that we sent him to grow. So it gives you an idea of some value. So if you remove everything from the plant so I can't identify it, I have a chemical test. But the other one we moved into with Damon Little was, and Chodan Seth actually, and Chelsea Specht was involved in some of this too, is that to develop DNA barcodes. So we have all these different plants, and what we wanted to do is find DNA barcodes for particular genera like the chemistry, but also species. And part of the reason for that was to identify their source and actually what species it was when they were lying, because that would tell us where they'd smuggled them from. So you can do those barcodes, and just a simple way of looking at that. But if you look on the left, you'll see that there's parts of the barcode there in diunes that segregate diune from all the other genera in there, the New World ones. And it also segregates, if you look at the weird pink colors and stuff, that segregate the different species of diune. And the same thing is present for Ceratozamia, had to put it in for those and the rest. So we have a good tool for conservation. So I want to do a little summary now. So I haven't talked about systematics very much, but what point I'd like to get across is that all of this different data plays back into the systematics, providing synapomorphies for the group, providing resolution inside for phylogenies, and allowing us to understand how and where they grow and why. Now, at the end of the day, I realized that the most important works that I do that have the longest lifetimes are my flora treatments and the Cycad action plan group that I was involved in for their conservation through time. And this all comes back into the systematics. And I think another thing that we need to tell the world right now is plants are not optional. No plants, no animals. Now, I can't thank the myriads of people for this, but I think you all know the person who's the most important person in this. And so I think I'm done and I'm ready for questions and a few little things you can read during questions. Thank you very much. Oh, I want to say one last thing. By the way, this is a progress report. This is not the final report. This is the progress report on Cycad biology. Thank you.
Dennis, thank you so much for that presentation. There's so much there, and I want to invite our audience now to come in with their questions. If you have a question, please go ahead and type it into the Q&A, and we'll go ahead and ask Dennis. We have one question coming in from Fabian. Do the bats themselves go crazy? If not, why is that?
Okay, I don't think the bats particularly suffer from it because the bats don't have a long enough lifespan like we do. Because for people, it accumulated through their lifetime, in a sense, you know, through a few years. And I don't know how long the bats live, but I always figured they don't live all that long.
Yeah, that makes sense.
Oh.
Oh, here's some questions. Oh, we have some reports. People just wanted to say thank you, and you did a fantastic job. Now, you talked a little bit about the glutamate receptors being in both plants and animals. Is there any effect on plants in the area, or is it mostly used to prevent predation?
So glutamate receptors are present, you know, they had that funny little gene tree, but they're present in bacteria, plants, and animals. And there are other plants that actually host these cyanobacteria, but they don't have the gold cell thing. They're just in the roots and stuff, and the stuff isn't excreted. So presumably it's only the cycads that should have a problem. The reason I bring this up because another group that has them is this genus Halagor. In the Halagoraceae, they have colonies of blue-green algae in their petioles. And there's a fern. Azolla has cyanobacteria, nitrogen fixing in their roots, and they're called green manure because they raise them in China in the rice paddies, which contributes to, they don't have to do so much nitrogen.
Excellent, thank you.
Yeah, can I just hop in for a second,
Of course.
Charlie? Yeah, Dennis, Alan Moreau wants to know, what is your favorite Cycad and why?
Oh, interesting. Thank you, Alan. I miss you. My favorite Cycad probably is Bauhinia, the genus Bauhinia, either one of the species. I find it just fascinating. Oh, I see chat boxes here. Or
Andrew would like to know, was Guam disease also found in other parts of the world where cycads were eaten? And he's thinking about Australian native peoples and the possibility.
Okay, yeah. So I do know the answer to that. So there's two other places in the world, well, three, I mean, other places where they're eaten. One of them was in the Caribbean, and they make zamia bread. And the other is the Aboriginals in Australia, and actually people in South Africa. Now, and then in Colombia, they actually have a poem to the plant they call the Zamius chigua. And they have a poem to it that during bad times, they would eat them. We had a woman who was a babysitter for us when the kids were little in Manhattan from Jamaica. And she said, oh, I know that plant. we used to have those during World War II for famine food, but I'm getting somewhere. There was a study done in Australia where they look at the concentration of both of the compounds in the plants, and then they looked at the concentration of what the people ate. And what the people do is they grind it up and they flush it, much like cassava is done in Brazil, for example. And so they looked at the effluent, and they discovered that the Aboriginal people got 100% of the bad compounds out of the plants before they ate the flower by washing it. So it didn't get to them. And I think that a similar thing was done in the Caribbean. And also the Caribbean people, I think, also would make slices and lay them in the sun, and a little bit of a black fungus would get on there and apparently change things around, and then they would wash it.
Great. Do the bats have any predators aside from people that have been studied for these impacts of toxicity?
I don't know. It's too late.
That's true. Those
But I
answers.
mean, I don't know. I assume they probably did, but again, the predators might not have long lifespan. I don't know what could catch a bat on Guam, but probably something besides people.
Several folks have asked about pollination in cycads and whether this is mostly beetle or wind pollinated. I think you mentioned that a little bit in the talk.
Okay, so as near as I can tell, there are no wind pollinated cycads, and there's been a lot of work. So after the original paper, there was a lot of work. And so there's a great body of literature out there, some of which is, I guess, summarized in Norstock's book, but actually since then, Irene Terry is one, and Shayla Saltzman's been working on that now, and various people, Willie Tang, and they've shown that there's Pharaxinotha, which is another sort of weevil type thing, and there's thrips. There are flies that do the pollination. The cones actually, many of them are thermogenic, so they produce heat. Remember I said that one, all the weevils came out of the cone overnight? So you can smell the cones. So there's thermogenic things that go on to attract them. And they actually go to the ovulate plants, but they don't feed on them. And I don't know why thrips do it. And in my picture, I didn't point it out, but I had a picture of pollen, and pollen actually looks like gold cells. So pollen itself is probably toxic.
Susanna wanted to say thank you, and this was a fascinating lecture, and loved the Benia example in particular. Do you have any other similar tips for paleobotanists on where preservation may be playing tricks on us?
Well, you know, I've actually published quite a bit with my paleobotany friends in Argentina. And when I go to BSA meetings, I always go to the paleo sections all the time. And I love to show that slide to them, of course. And, you know, you get the usual boos and hisses, but it's a good way, I think, you know, so-called neobotanists and paleobotanists working together, particularly people in anatomy morphology, make good combinations, because often an anatomist might recognize something that's distorted. But the one that always gets me is the stuff that Kevin Nixon and Bill Crepay and people are working on, this flora in New Jersey. And all the flowers are like, I don't know, 1/10 the size of the extants, but yet they're totally recognizable, which means under certain processes, you must be able to get some kind of uniform shrinkage where everything stays intact. So the fact of preservation, you know, it dries out, then it's preserved. So paleo is great though. The older I get, the more I identify with fossils.
Thank you, Dennis. We've had several people talk about how your work has really influenced their teaching of plant morphology over the course of years. Kathy Paris mentions that in her message. And just so thank you again for the community, Cycad specialists and botanists out there. Douglas asks, is the alpha taxonomy of the Cycads resolved, or is there still a lot of work to be done?
I'll tell you something. Damon did a little graph once about there were less than 100 known species. And the way I got into taxonomy, I was at Fairchild on my postdoc with Barry Tomlinson, but you're supposed to do things. And And so I decided to do something with cycads, but no one could identify any of them. That's actually how I got into the taxonomy of it. And not from me, but from other people too. Ken Hill, Michael Clonhay, many other people, Androvovides, et cetera. There got to be this big interest in them. And the number of species is now around 335. And that's all due to the push of exploration. It isn't going around and finding, 2 sheets and deciding one's a different species or whatever. It's fieldwork and exploration that was pushed by things like Flora Neotropica, Ken Hill doing all of Australia, me getting to go to China, all these people that did all of this work. And so I think, and we're still, I was involved in the description of, like, in the past 3 or 4 years, of like 5 new ceratozamias, several new zamias, but we're getting there. The alpha work is, the collections we're getting there. I think we're much closer. There probably isn't going to be a huge amount of addition, except maybe from some remote areas that, Colombia might be good where you couldn't go because of the grilleas. That was always, I went there once and that was not a good experience. I mean, that one. But so it's not done, Douglas. So I guess I just need to go out in the field again.
Once we're all traveling again.
Yeah.
Yeah, we've had several questions about the endangered status of cycads. I'm curious why that is in general. Why are so many cycad species considered endangered species? And then some folks have asked about endangered Mexican cycad species, and if you know anything about their situation.
Yeah, so they're endangered for, there are 2 major reasons. One is this over collecting that went on there. When I described Ceratozamia norstagii like an idiot, I put the locality in the paper, and within 6 months, 2,500 plants were taken from the wild. And there's only 2 or 3 populations, so you know what that means. So most of us quit putting precise localities down and have tried to block it in the herbarium because that's where these guys get it. And they get containers full. So there's that. And then habitat destruction. And they have long life cycles. There are some cycads that may take 20 years before they go from seed to cone. So you don't get many generations in a century. And the best example, I think, is in Mexico, was Zamia inermis, which Andrew Vovides described, from a single mountaintop in Veracruz. Lots of nice big plants, great. And the fields below it they spray insecticides on the agriculture. So when the planes do the aerial thing and they get towards the mountain, they pull up and they dump some of it on there. It ends up the weevils were wiped out, and the demography of the population was no plants under 20 years old or something like that. Sort of survives in botanical gardens because when those male cones break down, those sporophylls with the diapause weevils are in the crowns. So some of them, we got the pollinators in gardens. But I think habitat, there's too much worry about species and not enough worry, in my opinion, about habitat. And that's really where it comes from, is habitat destruction.
We've had several people curious more about cycad reproduction and the role of pollination versus seed dispersal in the historic or current distributions of cycads that exist.
There's very little, but there's some data on dispersal. The seeds are too big for most birds and things. For example, the mockingbirds in Florida try to take some plants, but they can only get a couple feet away because what do they call it? The gape, how far their bill can open for how long, sort of precludes it. However, there appears that there are a turkey-like bird that may disperse the seeds because you can eat the fleshy outer layer and regurgitate the seed or pass it through and you don't die. So hornbills do that in South Africa. There are some rodents where they found rodents in China and in Mexico that have moved seeds, but it doesn't appear to have long distance dispersal mechanism or a really effective way of doing it, you know. I'm sort of saying I don't know that there is much, but there are some data out there that do that.
You've talked about cycads in different parts of the world. Is there a particular center of diversity of cycads? And how are cycads doing in terms of their conservation status now?
There's the diversity is really, really interesting. So one real center of diversity is Mexico. It has 3 genera, okay? Australia has 3 endemic genera, and then South Africa has 2 endemic genera. So there's all this endemism that follows really a Pangenian distribution when you put the fossils in. Cycas is widespread throughout the Pacific and through Asia and Australia. I think their conservation status, since I've been working on them, I think there's more attention drawn to it. And there's 2 things right now that I think are being effective. One of them was something that was put together originally, was funded by the IUCN or the Cycad Specialist Group. And that's in Mexico, where Andrew Ovides set up nurseries with indigenous people who wouldn't. So what I'm saying is some gringo would go down there and give a guy 6 pesos for a plant and sell it for $200 here. Well, that wasn't a very good system. And so the people shut down doing that and started raising plants from seed in nurseries. And the bigger the plant, the more the value. So that became 1, no habitat destruction, 2, protection of the plants, and 3, an economic basis for people. So that's been a successful thing, I think, in Mexico. The other thing that's going on is some institutions like Montgomery Botanical Center and others have big nurseries now, and they try to grow things from seeds, because you can move seeds fairly easily, and try to get, several plants, more than 12 plants of a species that can represent some male plants, some female plants, avoid siblings, to have genetic diversity so that they can be reintroduced into habitats where they were removed through overcollection in particular. So there's progress.
A lot of folks are curious about what's next. What's the future of Cycad research? What are you going to do over the next 40 years of your career? What's the most important kinds of questions that people are asking and need to be answered?
Well, the project now that I'm doing with Michael Colonhay in the Maine is that we need to produce an online monograph. There's online now, there used to be the Cycad pages, which are defunct, and there's now the world list of Cycads that Michael does. And so we would like to have all the taxonomy up there for all the species, all the nomenclature, the descriptions, photographs. I want to put in all the historic material I have, like Chamberlain's slides and his pictures from the field in 1912. So that's one kind of thing. The other thing that's going to go on, I think now, is beginning to look at a functional plant anatomy and physiology, right? How do they do things to survive? And there's young people. Shayla Salzman is one of these young people. Michael Colonhay is another. There's Mario Caorio. Young people coming along that are going to build on the things that their predecessors did, just like I built on what Knut Nordstog did and what Charles Joseph Chamberlain did. So I think it's in good hands. And to be quite frank, I can't predict it, because who would have ever thought I would be like, sucking genes out of cycads? It's like, what is next? Probably the AI world is going to get in here. So I think there's, it's looking great.
That's great. And we all have a part to play.
Exactly.
What role do you see for conservatories and botanical gardens for cycad conservation? Is it more educational or production and propagation of materials for plantings?
Okay, I'm very pleased about that. That's a very interesting thing. And I've actually sort of occasionally talked about the ex situ, in situ conservation in cycads, and always said, you know, what's the point of ex situ? Because if a habitat's gone, how are you going to do it? I've changed my mind on that, you know, as obviously in my previous answer. But one of the great roles of these collections, and I could not have done that anatomy and chemistry and all that stuff without the living collections at Fairchild and later at the Montgomery Botanical Center. And you know what they do? They provide these plants that we can study to understand their biology and then apply that to the environmental situation. We discovered insect pollination in cultivated plants because the bug happened to come in the crowns of these zamias that had been growing there for 20 years. So that's a real value of those. And in fact, if Patrick Griffin is watching, I actually need an ovulate emerging stem of any cycad species that you can spare. So beware when I come to Miami.
We have some questions about the ICUN Cycad specialist group.
Yeah.
How did it form? What was your role in that group? How did you interact with CITES, and what are the next steps for that group?
Okay, so they formed, I think Cynthia Giddy got the IUCN specialist group started. I think she was the first chair. And then I think Knut Norstog was the second chair for us, but I did it for like 15 years. And I also went to CITES meetings for a while to represent the community. And I want to explain this a little bit because there were a lot of knee-jerk people on both sides of the issues. The issues was, no, you can't collect one, you can't bring it in, blah, blah, blah. They made the permit for a single seed the same as a whole female plant, which might take 100 years of reproduction out. And so I tried to bring together the extreme communities of collectors who said, I don't care, just take them all. We better get them now before they go extinct, you know, to the guys who didn't want anything moved. So we tried to constitute the committee of interested parties from different points of view. And that has led, I think, under John Donaldson's leadership to moving everything forward in terms of the biology of the plants, trying to utilize that and try to incorporate those things into the way we view protection and the way we try to get behavior. One of the behavioral changes that's gone on, I think, has been, you know what, it's really cool if you get some seeds and grow a plant for 5 or 6 years and start distributing seeds from that. You know, that's the cool thing to do rather than bringing mature plants from the wild. So there's a lot of education to the public, I think to the scientists and to the horticulturists, and they're all cooperating together for the good of the plants. And that's the way it's going to keep going with Patrick running it now and with John. And John Donaldson as the chair of the committee, it's really interesting because he's not a botanist. He's actually an entomologist. But he was totally into the pollination biology and all those kinds of things and the ecology of the plants and the phytogeography. So I think that also gives an example of different kinds of people working together for positive ends.
That's fantastic. And I think that's a great place to leave us. There are actually so many questions, way more than we have time for today. Dennis, your work is really inspiring for so many people, and I'm sure those folks will love to follow up with you later on.
They all helped me. They all helped me. You're all part of it. Thanks a lot.
Yeah, thank you.
Mm-hmm.
And thank you so much to our audience for being here today and for joining us. If you missed any part of this event or want to watch us again, a recording will be saved and it will be shared online on the New York Botanical Garden lecture library. And once that's published, everyone registered here today will receive a link. We hope you'll join us for future science, conservation, and humanities lectures from the New York Botanical Garden. On Friday, November 6th, we have experts discuss rare plants in our local area and what we can do to help protect and understand them. Then on Friday, November 13th, we'll bring together Indigenous environmental experts from the United States and Canada to discuss how Native communities are researching and addressing threats to their lands from extractive industries, pollution, and climate change. On Tuesday, November 17th and 24th, we'll take a global perspective on the biodiversity crisis and invite scientists from around the world for a two-part symposium on the topic of plant extinction. Stay tuned for invitations to all of these events in your email. And for those who haven't already registered for our mailing list, I'll share a link in the chat in just a moment. Until next time, please stay well, stay green, and take care. And thank you, Dennis, once again.
Bye.